Hello everyone, myself Katrina. Welcome to the next class of digestion and absorption. So in our previous classes we have discussed about different organs in the alimentary canal. We have also discussed about different glands present in the alimentary canal. So in today's class we are going to discuss about the different enzymes secreted by these glands and how these enzymes help in the process of digestion. So the process of digestion is accomplished by mechanical and chemical processes. Okay. So now we will see the digestion processes in each organ of the alimentary canal. So let's start with the buccal cavity, where two major functions occur. One is mastication of food, another one is facilitation of swallowing. Okay. So the mastication is done by teeth, tongue and saliva. When the food gets mixed with saliva with the help of tongue and the teeth break the food and grind the food, means the overall chewing of food is known as mastication. After that, the mucus in saliva helps in lubricating and adhering the masticated food particles and make it a bolus. Okay. Then the bolus is pushed towards the pharynx for deglutition, which is also known as swallowing. Okay, deglutition and swallowing are same. Okay, then the facilitation of swallowing means the passing of bolus in the esophagus from the buccal cavity through pharynx. Okay, so the food is pushed back against the epiglottis. If you remember epiglottis from our previous classes, that during the swallowing of food, the epiglottis close the windpipe. Okay, so when the epiglottis get closed, the esophagus opening become wider. Okay, then the bolus passes down through the esophagus by a wave-like movement or wave-like constriction in esophagus, which is known as peristalsis. Okay, now we will see the secretions of salivary glands. Salivary gland secretes enzymes and some electrolytes. Enzymes, we have studied that the main enzyme in salivary gland is salivary amylase. Okay, and another enzyme is also secreted by salivary gland known as lysozyme, and it acts as an antibacterial agent, means it prevents infections. Okay, and the electrolytes secreted from the salivary glands are sodium, potassium, chlorine and bicarbonate. Okay, so the function of salivary amylase. Salivary amylase breaks the starch. Starch is a type of carbohydrate. Okay, it is found in rice, potato. Okay, so 30% of the starch it gets hydrolyzed, means it gets breakdown in the oral cavity or the buccal cavity by the action of salivary amylase. Okay, means salivary amylase help करता है starch को break down करने में, and break down करके क्या form करता है, maltose form करता है. Okay, so ये कहाँ पे हो रहा है, ये होता है buccal cavity में और oral cavity में. Okay, the pH of salivary amylase is 6.8. So it is fixed. You have to remember this. The pH of salivary amylase is 6.8. Okay, and what does the salivary amylase do? It breaks the starch, which is a type of carbohydrate, into its simpler form, which is maltose. Okay, though maltose is not the simplest form of the starch, और भी ब्रेक डाउन होगा माल्टोज का, बट ये जो फर्स्ट इनिशियल ब्रेक डाउन है स्टार्च का, ये होता है ओरल कैविटी में विद द हेल्प ऑफ और विद द एक्शन ऑफ salivary amylase. Okay, and lysozyme is an antibacterial agent. Okay, then the food will move to the stomach, means in the next organ, because माउथ के बाद फेरिंग्स एंड ओसोफेगस में कोई डाइजेशन प्रोसेस नहीं होता है, तो फूड डायरेक्टली देन स्टोमक में जाके ही डाइजेशन होगा. ओके, क्योंकि माउथ में सिर्फ डाइजेशन होता है कार्बोहाइड्रेट का, मीन्स और स्पेसिफिकली अगर हम लोग बोले स्टार्च का डाइजेशन होता है, वो भी 30% ऑफ द स्टार्च का. ओके, सो इन स्टमक the food stores for around 4 to 5 hours. Okay, and we know the sphincter, which is gastroesophageal sphincter, it controls the passage of food into the stomach from the esophagus. Okay, so in stomach what happens, food mixes with the acidic gastric juice by the movement of the muscles, means the stomach muscles. Okay, and it makes it semi-digested, acidic because there is acid present in the stomach, acid HCl. Okay, and it makes it pulpy mass, and it is known as chyle. Okay, the HCl and the enzymes of the gastric juice, they all helps in the chemical simplification of food. They all helps in the breakdown of food. So the secretion of gastric glands are HCl, of course, which is hydrochloric acid, and pH is 1.8, and the enzymes, there are different enzymes which are secreted in the gastric gland: pepsinogen, prorenin, and lipase. The main enzyme is pepsinogen. Okay, and this prorenin, it is found only in infant. सिर्फ बच्चों में ये एंजाइम्स मिलता है, प्रोरेनिन, एंड लाइपेज भी बहुत ही कम अमाउंट में बॉडी में मिलता है, ये भी मेनली बच्चों में मिलता है. तो इन एडल्ट ह्यूमन द मेन एंजाइम ऑफ गैस्ट्रिक ग्लैंड इज पेप्सिन. ओके, एंड HCl, it helps in the activation of the enzyme because this pepsinogen, prorenin, they are in inactive form. Okay, so let's see the function. So pepsinogen is in inactive form. It gets activated with the help of HCl. Okay, HCl help करेगा pepsinogen को to convert it into pepsin, which is the active form. Now the pepsin will breaks the proteins into proteases and peptones. So these are the broken form of proteins. Next is the prorenin, which is the inactive form of renin, and this prorenin gets activated into renin with the help of HCl, and it is found only in infant to digest the milk protein. That's why it is known as proteolytic enzyme. Proteolytic enzyme means the enzyme which helps in the digestion of proteins. Okay, then lipases, they are also secreted in very small amount, and they acts on emulsified fats, means they helps in the breakdown of fats, and in infant they helps in digesting around 25% of milk fat. Okay, means the infant when they consume milk, the milk consists of proteins and fats. The proteins get digested with the help of renin, and the fats are digested with the help of lipases. Okay, so along with these secretion, the gastric gland also secrete mucus and bicarbonates. Okay, so these mucus and bicarbonates, they protect the mucosal epithelium of stomach from the highly acidic pH. Okay, so mucus होगा तभी जाके जो stomach का wall है, it will be protected from the acid. Okay, otherwise जो HCl है उसका pH है 1.8, तो जो stomach है वो जल जाएगा. तो to protect it from burning by the acid, mucus and bicarbonate, they means prevent the stomach. Okay, after stomach, the food will now move into the small intestine. In small intestine, there are secretions from 3 different organs or glands. Okay, so the first one is from the liver, which is the bile. Then from the pancreas, the pancreatic juice will be released into the small intestine, and then the intestinal juice, which is secreted from the small intestine itself. Okay, so now let's see the function of these secretion one by one. The first one is bile juice, which is secreted by the liver. Okay, this bile contains bile pigments, which are bilirubin and biliverulin. Along with bile pigment, there are bile salts, cholesterol, and phospholipid. Okay, so these are the secretion of the liver. Okay, so the bile helps in emulsification of fats. Okay, means it helps in the breakdown of fats, and also it activates lipases, which is another enzyme. Okay, next is the enzymes in pancreatic juice. ये जो पैंक्रियास से उससे जो एंजाइम प्रोड्यूस हो रहे हैं वो भी डिस्चार्ज होगा इनटू द स्मॉल इंटेस्टाइन. So the enzymes which are present in pancreatic juice are trypsinogen, chymotrypsinogen, procarboxypeptidase, amylase, lipase and nucleases. Okay, so among them the trypsinogen, chymotrypsinogen and procarboxypeptidase, these are the inactive form. Okay, then the trypsinogen will be converted into its active form, which is the trypsin, with the help of an enzyme enterokinase, which is secreted by the intestinal mucosa. Okay, trypsinogen, which is an enzyme of pancreatic juice, it is an inactive enzyme. It converts into its active form, which is trypsin, and in this conversion the enzyme enterokinase helps, which is secreted by intestinal mucosa. Okay, now the trypsin will activate other inactive enzymes. It will activate chymotrypsinogen into chymotrypsin and also procarboxypeptidase into carboxypeptidase. Okay, now let's see the function of the different enzymes which are present in the pancreatic juice. Okay, so first of all let's see the about the breakdown of proteins. So the pancreatic juice, trypsin, chymotrypsin and carboxypeptidase, they helps in the breakdown of proteins or peptones or proteoses into dipeptides. Okay, protein अगर पहले कुछ ब्रेक हुआ है like with the help of pepsin, अगर वो ब्रेक हुआ है then it will get converted into peptones and proteases. Okay, अगर कोई प्रोटीन ब्रेक नहीं हुआ है तो वो प्रोटीन्स रहेगा. ओके, सो कोई भी फॉर्म का प्रोटीन हो जो प्रीवियसली ब्रेक डाउन हो चुका है उसको फिर ब्रेक डाउन किया जाएगा विद द हेल्प ऑफ ट्रिप्सिन, काइमोट्रिप्सिन एंड कार्बोक्सीपेप्टाइडेज, और इसको ब्रेक डाउन करके इट विल बी कन्वर्टेड इनटू डाइपेप्टाइड्स. ओके, नेक्स्ट इज ब्रेक डाउन ऑफ कार्बोहाइड्रेट्स. ओके, सो कार्बोहाइड्रेट्स पहले एक बार ब्रेक हुआ था इन द बकल कैविटी विद द हेल्प ऑफ salivary amylase. ओके, और सिर्फ एक पर्टिकुलर टाइप ऑफ कार्बोहाइड्रेट ही ब्रेक हुआ था, दैट इज स्टार्च. वो भी 30% ऑफ द स्टार्च. ओके, सो अदर देन स्टार्च, और जो स्टार्च भी जो 30% के अलावा जितना 70% ब्रेक डाउन नहीं हुआ था, जितना भी कार्बोहाइड्रेट है they will now break down into disaccharides with the help of the enzyme amylase, which is secreted by the pancreas. Okay, next is the breakdown of fats. Also the fats which are present in the food, they gets break down with the help of the enzyme lipase. Okay, which is activated with the help of bile, and this lipase converts the fats into diglycerides and further it converts it into monoglycerides. Okay, also the breakdown of nucleic acid occurs in the small intestine with the help of the pancreatic juice nucleases. Okay, it breaks the nucleic acid into nucleotide and further it breaks it into nucleosides. Okay, so here you can see the how important the pancreatic juices are, because the pancreatic juice are breaking all different types of nutrients, be it proteins, then carbohydrates, fats, nucleic acid. Okay, but अभी तक जितना ब्रेक डाउन हुआ है ये कंप्लीट ब्रेक डाउन नहीं है, मीन्स एकदम सिंपलेस्ट फॉर्म ऑफ ब्रेक डाउन नहीं हुआ है, और another breakdown is left जो ब्रेक डाउन होगा with the help of the enzyme secreted by the mucus of the small intestine. Okay, so now see the enzymes present in the small intestine, which are also known as succus entericus. Okay, so succus entericus, as I had previously told you, that these are a group of enzymes. Okay, so So the enzymes which are present in succus entericus are disaccharides, dipeptides, lipases and nucleosidases. Okay, so these enzymes act on the end product of all the reactions. जितना भी बॉडी में ब्रेक डाउन हो रहा है या जितने भी डिफरेंट ऑर्गन्स में ब्रेक डाउन हुए हैं, ये जो सकस एंटेरिकास में जितने भी एंजाइम है, दिस विल एक्ट ऑन द मींस द ब्रोकन फूड and it is the end product. They will form the end product. Okay, after that the digestion process will be over and the absorption process will start. Okay, so these final steps in digestion, they occur close to the mucosal epithelial cells of the intestine, because after that the absorption need to occur. Okay, so let's see the function of enzymes present in succus entericus. The first one is dipeptidases. Okay, so the dipeptidases will act on dipeptides, which are the broken form of proteins, and convert it into the simplest form of proteins, which is the amino acid. Okay, means the dipeptidase will convert the broken form of proteins, means dipeptides, into amino acids, the simplest form of proteins. After that, this amino acid will be absorbed into the body. Okay, next is the action of disaccharides. Disaccharides, the maltase, lactase, sucrase, these are all disaccharides. They act on disaccharide. Okay, maltose is a form of disaccharide. Similarly, lactose and sucrose are also disaccharide. So maltose will be converted with the help of maltase into glucose and glucose. Okay, similarly, lactose will get converted into glucose and galactose with the help of disaccharidase lactase, and sucrose will be converted into glucose and fructose with the help of sucrase. Okay, means maltose, lactose and sucrose, they are disaccharide, and the enzyme which breaks the disaccharide are disaccharidase, which are sucrase, lactase, maltase for maltose. Okay, and they convert into, or they digest it into, the simplest form of carbohydrate, which are glucose, galactose, fructose. Okay, next is the enzyme nucleotidase and nucleosidases. Okay, so nucleotidase will act on nucleotides and convert it into nucleosides. Okay, and nucleosidase will act on nucleosides and convert into sugars and bases. Okay, means nitrogenous bases. Okay, similarly, the enzyme lipases will act on monoglycerides and diglycerides and convert them into fatty acids and glycerol. Okay, so these amino acid is the simplest form of proteins. This glucose, galactose and fructose, these are the simplest form of carbohydrates. The sugars and bases, they are the simplest form of nucleic acid. Fatty acid and glycerol are the simplest form of fats. Okay, means the enzymes which are present in succus entericus, they convert the semi-digested food into the simpler forms, after which they get absorbed into the body. All these reactions, they occurs in the duodenum region, that is the first region of the small intestine, after which the simpler forms are absorbed in the jejunum and the ileum region of the small intestine. Okay, so that's all about the breakdown of food materials into simpler ones, and these breakdown occurs with the help of different enzymes secreted by different glands and different organs of the alimentary canal. Okay, so that's all for today. Next day we will continue more of this chapter. Okay, thank you.